Okay, so yeah, let's get started. Hello, everyone. Welcome to the Environmental Research webinar. I am Song-Kyun Park, faculty member in epidemiology and EHS. So this seminar series is sponsored by the Michigan Life Stage Environmental Exposures and Disease Center, also known as M-LEEaD. I am very delighted to have today's speaker, Dr. Brittney Baumert. So Dr. Baumert is currently a postdoctoral fellow at the University of Southern California Keck School of Medicine. She earned her PhD in Environmental Health Sciences from Emory University, an MPH in Epidemiology and BS in EHS at the University of Georgia. And so right before we begin this webinar, I learned that Dr. Baumert will join University of Georgia as a tenure track faculty member next summer. So congratulations. Dr. Baumert research focus on precision environmental health, specifically how persistent organic pollutants, particularly PFAS, contribute to metabolic disease risk in children and adolescents. She integrates epidemiology with multiomics and translational approaches to uncover mechanism linking persistent organic pollutant exposure to metabolic disease with the goal of identifying biomarkers and advancing precision strategies. So please join me in giving a welcome to Dr. Baumert.
Hi, everyone. Thanks so much for that very nice intro. It's nice to join you all today. And so I'm excited to talk about PFAS and metabolic health among a cohort of adolescents. So approximately 1 in 5 US children and adolescents have obesity. And we know that certain subgroups are more impacted than others. So Hispanic and non-Hispanic Black children, as well as families from lower incomes, tend to have greater rates of obesity. And you can see on this map here from the CDC, it's showing the percentage of high schoolers with obesity in 2019. And so these percentages are quite high. You can also see this kind of cluster in the Southeast. And we know that there are a number of adverse health impacts associated with obesity. We know that there's adverse effects on metabolic and endocrine, as well as bone development, and that the chronic pro-inflammatory state contributes to really long-term disease risk. So there's negative effects also on the psychosocial well-being and mental health of adolescents with obesity. And these health implications track into adulthood. So children with obesity are 5 times more likely to remain obese in adulthood. So really this highlights that there's need for early prevention and intervention. We've seen the rising use of bariatric surgery among pediatric and adolescent populations. So bariatric surgery is quite an effective treatment for severe obesity, as well as the related comorbidities such as insulin resistance, dyslipidemia, MASLD, or metabolic dysfunction-associated liver disease. You may know this as NAFLD. It was recently renamed to MASLD, and hypertension. And so early weight loss intervention before adulthood, we know lowers the future risk-related adverse health effects in adulthood. Kind of one of the concerns around bariatric surgery is this remission that we observe following bariatric surgery. So in this figure, I'm showing BMI among adolescents in the Teen Lab study, or the Teen Longitudinal Assessment of Bariatric Surgery, which is what I will use to present today. But yeah, so we see that there's this kind of steep decline in BMI following bariatric surgery, followed by this kind of upward trajectory. You also can notice that there's quite a big heterogeneity and success following bariatric surgery. And so there's a lot of questions around what factors influence this heterogeneity. We also observe remission in type 2 diabetes, hypertension, and dyslipidemia following bariatric surgery, but noticeably more so, we see this big heterogeneity again, noted by the 95% confidence intervals in success of treatment. And so I've really been interested in looking not just at metabolic health among adolescent populations, but also remission following bariatric surgery and factors that may help explain this heterogeneity that we observe. We know that there exist key risk factors such as diet, sedentary lifestyle, genetic susceptibility, just to name a few. But we also know that environmental exposures, particularly endocrine disrupting chemicals such as per and polyfluoroalkyl substances or PFAS, are associated with metabolic health. And this is referred to as the toxicant metabolic health hypothesis. And notably, it's a modifiable risk factor. So after exposure, PFAS can accumulate and persist in the human body for years. They've been detected in the blood of greater than 99% of the population in the US. And there's really widespread health effects. And so you can see in this figure that PFAS really affect almost every organ system in the human body. They impair immune function, disrupt endocrine systems, and increase the risk of metabolic diseases and cancers. So what are PFAS? PFAS are a chemical class of thousands. I've seen reports up to more than 14,000 now chemical congeners. They've been widely used in industry because of their persistence. So they stick around forever, both in the environment and biologically speaking, which is why they've been coined the forever chemicals. And as I said in the last slide, they've been detected in the blood of almost everyone in the US. And so we're exposed to PFAS through water contamination, through nonstick cookware, food packaging, containers, cosmetic and personal care products, as well as textiles such as outdoor gear. And you can think of those water-proof clothing. So rain jackets oftentimes have PFAS on them. Water is a primary source of exposure to PFAS in the United States with over 9,000 known contamination sites. And so this image is from the Environmental Working Group, and I think it does a really nice job just highlighting how widespread this problem of PFAS exposure is. It's been shown that over 200 million people may have contaminated drinking water. And so kind of coming back to the model of bariatric surgery, it's really an ideal kind of model for investigating the effects of PFAS on metabolic outcomes for a number of reasons. So one of those reasons is that we see this sharp time-anchored physiological shift with dense longitudinal phenotyping. We're able to do within-person comparisons. And then there's also the opportunity to study tissue blood relationships, which has really been key. It's pretty hard to get liver biopsies from people. They're often not really understandably so open to that. But in a bariatric surgery model, it's actually much easier to get adipose tissue or liver or other tissues beyond blood. It's also for adolescents, it's a sensitive life stage. So we see this rapid growth and endocrine remodeling that may modify PFAS susceptibility and cardiometabolic trajectories. And then we have these built-in endpoints that are relevant to PFAS biology. So weight loss and regain and lipid metabolism, inflammation, liver disease, which are all also plausible PFAS targets, and primary outcomes within a bariatric surgery or post-bariatric surgery model. And so today we'll use the Teen Lab study, which is the teen longitudinal assessment of bariatric surgery study. And it's a prospective multi-center observational study of adolescents that are under the age of 19 years of age and underwent bariatric surgery between 2007 and 2012. We enrolled participants at participating clinical centers in the United States, and it includes 214 adolescents. Notably, we have 64 participants with both plasma and liver biopsies, and we have a lot of omics data measured. So we have longitudinal metabolomics, proteomics, and lipidomics up to the 36-month time point, And then we have plasma PFAS measured at baseline up into the 36-month time point, and then medical record data abstracted for each of the time points. And so of the 214 participants in the parent teen lab study, our work includes 186 individuals that had both PFAS measures as well as outcome measures. And so Teen Labs is predominantly female, white, with a mean age of 17 years at baseline, and come from families with less than $75,000 a year. And so, So this bariatric surgery model has really allowed us to look at the implications of a huge weight loss scenario on circulating PFAS concentrations. Previous studies have shown that donating blood and breastfeeding have decreased circulating PFAS. But within our study, we're able to look at this in the context of a large weight loss event to a longer time frame, so up to 5 years post-op. And we observed that there's a sort of bariatric surgery effect among PFAS. So using estimated marginal means for longitudinal changes in plasma PFAS following surgery, we took into consideration the temporal and within person trends in PFAS. And we see that there's this kind of initial plateau, or it's kind of a static observation, followed by a sharp decline from the 6 to 36 months following bariatric surgery. And then the bariatric surgery model also allowed us to investigate the plasma liver specific PFAS concentrations, as well as the measured ratio of liver to plasma. And notably, we see here that PFOA and PFOS were measured in the highest plasma concentration despite phase out. They were still a major contaminant at the time of surgery, measured in plasma and liver. And then PFHPA has been interesting in all of my work, but really stood out in this exposure assessment that we did. And we observed really low levels of PFHPA in plasma and liver, but notably it had this very high liver to plasma ratio. So it was 26 times higher than PFOA. And so we followed this kind of very basic analysis of plasma and liver and the ratio of plasma to liver PFAS up with mass balance model. And for time, I'm not getting into details, but this paper has been published and I'm happy to take questions on it and really set the stage for our epidemiological research that we followed up with. So we were interested in creating a mass balance model to kind of predict the ratio of plasma and liver, and in thinking that it's really hard to get liver biopsies, but if we could provide this data, then others could predict liver concentrations. And so we were able to utilize this more complex toxicokinetic model to predict the liver plasma PFAS concentrations. And we observed that there's quite great prediction for the liver PFAS or liver plasma PFAS concentrations for most of our congeners, except notably the PFHPA was quite underestimated, which indicated that there was additional transport mechanisms beyond partitioning. And so it really is due to the fact that we had this resource of the bariatric surgery cohort that we could directly observe these findings. And we observed that liver is a key site of PFAS accumulation, and PFHPA showed quite unique toxicokinetic and quite unexpectedly high liver plasma ratios. And so all of this exposure assessment data results I share in that they set the foundation for our next steps, which were epidemiological questions related to metabolic disease outcomes. And so coming back to the framework of PFAS and metabolic health, we've been interested in the question of PFAS and metabolic health in the context of remission after bariatric surgery. And so while there's existing literature on PFAS and metabolic health, the scenario within bariatric surgery is quite unique. And additionally, there are a number of gaps and limitations in our current knowledge. So there's variability in biomonitoring of PFAS exposure, as well as kind of some challenges within model systems. So animal models often fail to fully recapitulate the human metabolic disease pathways. We know that human cohorts are quite laborious and there's long-term follow-up needed. And then in vitro models are often used as single cell type and can't mimic organ level interactions. And then there's also many limitations and gaps in our understanding of data integration and translation, as well as the chemical mixtures of PFAS. And so jumping into example one, in this paper, we were interested, it's been published in Nature Communications Medicine. It was just published a few weeks ago, but we were interested in PFAS and MASLD. And so initially we kind of started out with just this arm where it was going to be a quite basic paper of looking at PFAS and MASLD measured using liver biopsy. And then we decided to kind of expand it to include an in vitro component where we expose liver spheroids to non-cytotoxic doses of PFAS. But then we thought it would be really interesting if we could figure out a way to kind of integrate all of this. And so we did that to determine high-risk clusters of individuals for MASLD based on their protein, so proteomics, metabolomics, and PFHPA kind of clusters to identify those at high risk for MASLD. And so, like I said, as a first step, we did this very basic epi analysis where we looked at PFAS and MASLD using this histologically defined feature data. And notably, we only saw that PFHPA was associated with MASLD. We didn't observe associations with any of the other PFAS congeners. And so because we had this really robust data, we were able to follow up with some additional histologically defined MASLD data. And so we next investigated the concentration-dependent response relationship between PFHPA, and we observed that with increasing PFHPA exposure, we observed increasing severity of MASLD. And then we also observed this clear concentration-dependent response trend across quantiles of PFHPA exposure with MASLD. And then we followed up a bit further, and we observed this clear association between PFHPA and MASLD phenotype, seeing that with increasing exposure to PFHPA, we observed that it was associated with increased severity of each disease phenotypes. And so we had this kind of really consistent story with PFHPA and all of these different well-defined MASLD outcomes. So from there, we said, okay, let's do a deep dive on PFHPA and try to uncover some of the related mechanisms. And so we partnered with Lucy Golden and Ana Maria Garcia at USC, and they have this really nice liver spheroid model. And so we use these 3D liver organoids that mimic human liver function, and they include multiple cell types. So they have hepatocytes and Kupffer cells and endothelial cells. And we expose the liver spheroids to PFHPA at a non-cytotoxic dose of 20 micromolar for 7 days. And then we evaluated gene expression with single-cell RNA sequencing in the liver spheroids. And so those who don't know what a liver spheroid is, you can think of it as this like little mini liver, essentially. So next, we were interested in integrating the liver spheroid gene expression data with our human epidemiological data from Teen Labs. So using the Teen Labs data, we performed a PWAS and an MWAS with PFHPA. And then the differentially expressed genes data set from the liver spheroids that were exposed to PFHPA were integrated with the MWAS and PWAS data sets using the OmicsNet platform. And so we selected the most significant pathways and identified 19 metabolites in the gene metabolite overlap pathways and 6 proteins in the gene and protein overlapped pathways. And so this figure here shows the overlapping pathways of multi-omics associated with PFHPA exposure from the OmicsNet platform. And on the left, we see gene expression by MWAS results, and on the right, you can see the gene expression by PWAS results. And so we really observed alterations in lipid metabolism amino acid metabolism, activation of oxidative stress, and then also some pathways that were more associated with cancer. And so from here, as a final step, we are interested in using all of this information to integrate these features from overlapping pathways into a clustering method to identify teens at higher risk for MASLD. So we extrapolated the metabolites and proteins that we observed in each of these overlapping pathways from the liver spheroid differentially expressed gene data set. And so you can now kind of think of this as like our data dimensionality reduction step, but we did it by kind of a more informed selection process. And so these are those results of using a latent clustering method to integrate PFHP exposure with proteins and metabolites identified in the previous step to identify subgroups of adolescents at high risk for liver disease. And so this advanced risk prediction technique is also known as latent unknown clustering integration of multi-omics data, which is quite a mouthful. So we just call it LUCIT. And it was developed by Dr. David Conti and his team at USC to integrate multi-omics features with environmental exposure data. And so in this case, PFAS. And so we use MASLD as yes, no, and then the model simultaneously incorporates proteins, metabolite clusters, and adjusts for site, age, sex, race, and parental income. And so in this figure, the metabolites are in green and the protein profiles are in orange or that kind of yellowish color. And you can think of these as two separate clusters or two profiles. And I personally, I shown this with this figure, it's the same results, but I think it's depicted in a little bit more clear way, where you can see kind of the high risk cluster more clearly. So you can really, I think, see the two distinct multi-omics risk profiles, and the high risk MASLD profile is those with increased protein features and decreased metabolite features associated with increased exposure to PFHPA. And so notably, MASLD was 7 times more likely in this high-risk profile and shows proteins related to liver metabolic stress, inflammatory and fibrotic signaling, and coagulation changes, all of which we know are really closely linked to MASLD onset and progression. And so that was a lot, but so our take-homes from our first example is that plasma PFHPA is associated with MASLD severity, and we observed this really nice kind of concentration-dependent effect. And then from a mechanistic and multi-omics standpoint, we observed dysregulation of lipids, inflammation, and oxidative stress, as well as cholesterol biosynthesis pathways in relationship to exposure to PFHPA, and then identified this high-risk cluster that was 7 times with 7 times higher odds of MASLD. And I think the biggest take home of this study is, while we did find really interesting results, I think it highlights the power of team science, and kind of when you get out of your own little silo and start collaborating with your neighbors, it's really, I think it's really powerful in what you can do in a translational science setting. So for example, 2, we are interested in examining the relationship between PFAS and postoperative weight regain in adolescents. So we looked at baseline PFAS measured at the time of bariatric surgery and postoperative weight regain until 5 years post-op. And so this figure shows the median PFAS levels with 1 standard deviation above and below. And so we see at this lower concentration of PFAS, we see that adolescents gained about 16 kilograms over the 4-year post-op period, and then this increased to 21 kilos in this higher exposed group, which is meaningful. And so here I'm showing higher sulfonic acid PFAS congener concentrations that are associated with increased weight regain following bariatric surgery in this 1 to 5 year post-op period. And so we observed similar effects as we observed with PFOS, but particularly this was true for PFHPS. So at quite low levels of PFHPS, teens regained weight at a rate of 2.6 percentage points per year, and this increased to a rate of 3.8 percentage points per year at the higher concentration level, which is 46 times faster. And so these results really add to the growing evidence that PFAS exposure may interfere with long-term success of weight loss surgery by promoting faster weight regain, even at relatively low exposure levels. And so as a next step in this analysis, we looked at PFAS mixtures using quantile G computation. So when looking at PFAS as mixtures rather than individually, higher levels of sulfonic acid PFAS, including PFOS, PFHXS, and PFHPS, were all associated with larger waist sizes following up to the 5-year time point post-surgery. And so a modest increase in sulfonic congener exposure, so 1 quartile, was associated with about a 4.4 centimeter greater waist circumference, and PFHPS contribute the most to the observed increases in waist size, BMI, as well as weight regain. And so our take home from our PFAS weight regain following bariatric surgery analysis was that higher concentrations of particularly the sulfonic acid congeners, PFOS, PFOS, PFHXS, PFHPS were all associated with greater weight regain, increased BMI, as well as larger waist circumference in the 1 to 5 year post-op period. PFHPS consistently emerged as a strong contributor to weight regain outcomes following surgery. And we observed that at higher levels of sulfonic acid congeners, adolescents regain more body weight up to the 4-year time point, over the 4-year time frame. And so I think these findings really highlight the potential for PFAS exposure to impact long-term weight loss success in teens who've had weight loss intervention. I also think that it points to the need for integration of environmental health within a clinical setting to consider in obesity treatment and recovery. And then for the last example, I was interested in looking at the association between PFAS and postoperative glucose homeostasis. And so for our longitudinal models of PFAS measured at baseline and type 2 diabetes markers measured up to 60 months post-op, we observed that higher PFHXS levels were linked to faster rises in fasting glucose between the 1 to 5 year post-surgery period. So participants with higher PFHXS concentrations had nearly 3 times greater yearly increases in fasting glucose compared to those at the lower levels. And this suggests that there's elevated long-term diabetes risk among these participants. And then we also observed that higher PFHXS levels were associated with faster rises in HbA1c between the 1 to 5 year post-surgery period. So participants with higher levels of PFHXS had around a 60% greater yearly increase in HbA1c compared to those in the lower levels. So this also highlights that worsening long-term glycemic control and diabetes risk among these participants. And then for our mixture analysis, we observed that exposure to PFAS mixtures is associated with greater insulin resistance at the time of bariatric surgery. Specifically, a quartile increase of all PFAS was associated with about a 6.7 unit increase, which is a sizable increase since normal fasting insulin is often less than 10 microinternational units per milliliter. And so the key contributors to this mixture were PFHPA, PFHPS, and PFHXS for both fasting insulin and HOMA-IR. And so PFHXS was associated with greater increases in fasting glucose and HbA1c in the 1 to 5 year post-op period. And we observed that our findings indicate that PFAS exposure may be associated with worse insulin resistance and play a role in attenuating improvements in glucose homeostasis among adolescents following bariatric surgery. So I think kind of what's the big picture summary, tying all of this together. We know that PFAS exposure is consistently linked to metabolic dysfunction in adolescents, affecting liver health, glucose regulation, and weight outcomes following bariatric surgery. PFHPA, as well as the sulfonic acid congeners, so PFOS, PFH6S, and PFHPS, were all key drivers of lipid dysregulation, inflammation, and oxidative stress, disrupting pathways central to metabolic homeostasis. Mechanistic and multi-omics integration across epidemiologic and experimental models pointed to PFHPA interfering with bile acid, fatty acid, and cholesterol metabolism. And then adolescent bariatric surgery cohorts really offer this unique translational model to study PFAS and metabolic resilience during a big physiological and endocrine kind of change. And so exposure to PFAS appeared to really attenuate the improvements in weight loss, including metabolic markers such as fasting glucose and HbA1c that were expected to improve post-bariatric surgery. And so we observed that clear dose response with PFHPA and MASLD at the time of surgery, as well as there was also sulfonic acid congeners with markers of weight regain and type 2 diabetes. And so I think ultimately that these points highlight PFAS as environmental modifiers of weight loss intervention and metabolic recovery, and that there's really truly implications for precision environmental health within the framework of obesity intervention outcomes. And so with this last slide, I'd like to thank my many collaborators at USC, as well as some of the other universities that we've collaborated with. It's truly been a team science effort. My primary mentor is Lita Chatsi, and she's really the best of the best. She's been a phenomenal mentor, and also our newly funded Superfund research program that continues to make this work possible. So thank you so much for your attention.
Great. Yeah, this is really exciting research.
Thank you.
Yeah,
Thank you. Thanks so much, Robin.
So while we are waiting for additional questions, so for the audience, so if you have any question, please use the Q&A the menu on the bottom, and then enter your question. And Brittney, you can also, so because we are recording this, the talk, and so that I will be reading the question, but you can also see a question directly from the Q&A if you are able to open that box.
Okay.
Yeah. All right, so we have a question from Dr. Vasansa Padmanabhan that liver being very vascular, how much of liver PA, I think this is about the biopsy, and the PA levels coming from blood contamination vs tissue level.
Can I ask real quick, how do I open, so you said I can open up the questions as well?
Maybe because you are now sharing your screen, so that's why you may not.
Oh, okay.
The button, yeah. So.
Oh, the Q&A, I see now.
Yeah.
Okay, great. Wonderful. Yeah, so liver being very vascular, how much of the liver PA levels are coming from blood? Oh, yeah, that's okay. So, yeah, that's an interesting question. So we found that it was PFAS dependent. So for PFHPA, it really sequesters in the liver, where some of the other PFAS congeners, they tend to be circulating. But I imagine that it's all coming from the vascular kind of component to this.
So, Dr. Padmanabhan, the correct, so the PA, actually, it was typo, so it is PFOS.
PFAS?
Yeah, PFAS.
Yeah.
And then another question is that how many PFAS analyzed or characterized in this study? I think this is your teen labs.
Teen labs, yeah. So for the work today, I presented using what our targeted PFAS measures, and they were measured by Philippe Grandjean's lab. And so we have about 7, I believe, PFAS congeners. in that data set. We also had Doug Walker at Emory University measure PFAS, and so he has standards for I believe 8 or 9 PFAS from when we did this analysis, but obviously within his library has a lot more PFAS measured. So there's a lot of room to kind of expand even into the perfluorome or to look at a lot of additional PFAS.
I really I found your this bariatric surgery and then weight loss and the weight regain that study design, which is a really interesting one. And so the for like a physician and doctor, so this information could be very useful. So when they have a patient who want to do this bariatric surgery for the weight loss and then maybe measuring PFAS could be important step, or so that can be important message to the public that the reducing exposure could be important. But compared to PFAS versus probably other factor also impact this weight remission and then the individual variation of this weight gain. And then so how the impact of PFAS is it is the larger than any other factor, or can you make some comment about so how important PFAS in terms of this weight regain?
Yeah, so I think there's quite a few questions with that. Yeah, I think this is very important in a clinical setting, and I shared with you before we hopped on to our seminar that I spoke at Obesity Week, which is Obesity Society's conference a couple weeks ago, and the kind of response from physicians was really encouraging, and that several physicians kind of came up to ask how they can look at PFAS within their patients and maybe use that as kind of a low-hanging fruit, right, for intervention when you're talking about these huge weight loss scenarios, is can we reduce exposure to PFAS to help maybe improve response to treatment and improve overall metabolic health. And so that's really encouraging. I think that that's a great kind of example of the partnership that can happen with physicians. And when we talk about precision and environmental health, ultimately, that's the actionable component, right, is actually getting it to patients. And so that's really exciting. I think there's a lot of work to be done within that. And so certainly environmental exposures in this case are part of that. But I think the multi-omics piece is also really interesting, which kind of gets at your question as well related to weight regain following weight loss intervention. And so and the paper that was recently published in Obesity, we just looked at baseline PFAS and then longitudinal markers of weight regain following bariatric surgery. I'm really interested in following that paper up with kind of looking at a multi-omics paper where we look at baseline kind of multi-omics clusters to see that component related to weight regain. So not a PFAS focused paper, but multi-omics weight regain paper. Diet's also a huge part, obviously, of weight regain. And so Teen Labs doesn't have phenomenal diet data. I would say it's kind of lacking diet data, and I think it's a big limitation within the study. And so following up in another cohort with robust diet data, I think is actually quite important, which also could be a source of PFAS exposure. So it's kind of a really complex question.
Yeah, it is. What about like a physical activity? So
that too, for sure. Yeah. Now with everyone wearing these Apple Watches, I think it's a lot more accessible to get activity data. And I think that that's another kind of follow-up question. We do have some activity related questions, but it is not really well defined.
Okay. another interesting point that I found is that this PFHPA, which is really
Yes.
interesting, and I don't know if like other organ system and the other health outcome, because the PFHPA detection probably lower than P4 or P4s, and that's why the many study focus on P4, P4s, and then PFHPA has not been the focus of the research, and this is why we don't have much evidence to compare between this one. But so can you elaborate a little bit more about why liver, but not P4 or P4s, but the
HPA.
PFHPA? Why this is specifically like bound to the liver cell or the lipid metabolism, and so that is really interesting finding.
Yeah, I would say during my postdoc that PFHPA has been my most interesting finding, actually. And I have about 1 million questions that I hope to kind of explore in the future. But yeah, the why is a big question. And so, you know, I wonder if it's, so Jesse Goodrich is another researcher at USC and has actually recently submitted a paper where we find interesting results with PFHPA and kidney function. And so we've talked a bit about this, and I think it's where kind of partnership with our toxicology friends is important in that I think we can look at kind of a compartment model where we focus predominantly on PFHPA. We wonder if it has to do with a different like lipid binding or kind of, we historically think of PFAS binding to proteins, particularly albumin. And so is there a different kind of mechanistic behavior of PFHPA? And we wonder if that's related to fat within kind of the interplay of adipose liver, if there's something there. So yeah, that is, I think, a really interesting finding. And yeah, to your point, a lot of people aren't looking at PFHPA, but it has really driven a lot of our results. So I think it's worth, if you can look at PFHPA, I think it's interesting because we're seeing quite low concentrations measured in our study, but we still see these significant findings.
So I am working with the study of women's health across the nations, one study, and unfortunately we didn't include PFHPA in our initial panel of PFAS, and so that's why we do not know how much the PFHPA detection in this population is, but for future research, I think I want to include this PFHPA in the future research as well. So then, but related to this question, and so as you mentioned that now, over, the 10,000 or even 15,000 PFAS are manufactured every day. But since 2000, this legacy PFAS concentration declined at the population level. But this is just my personal, the thought, because I don't have the supporting evidence, but the legacy compound decline, but the total PFAS exposure level may not decline because the industry, they need the PFAS in their the industrial application, and then manufacturing consumer product for this persistency or oil repellent and water resistant product. So then you're finding that the long chain and this PFHPA and the long chain PFAS versus short chain PFAS, and these days the PFAS are replaced with a more shorter chain PFAS. And is this the right direction?
Yeah, so I will say the study's from 2007 to 2012. So, it's not necessarily old, but it's not today. And so when we do look at decreasing trends of PFAS, we see that in our study, the legacy PFAS decline as well. And so we tried to, when we looked at changes in PFAS across time, we, I showed the figure of kind of the static trend and then a greater increase following that 6-month kind of time frame. And so we took into consideration kind of the annual trends in PFAS, and still observe that there's this big decline. I also think it is a bit whack-a-mole, right, with PFAS, because they regulate one, but then there's 14,000 others. And so kind of there's questions around breakdown products of PFAS, and kind of what the parent PFAS might be, or it's just this big PFAS soup. And so I do think that we need to be thinking about these questions of where are ultimately what is the parent PFAS, what are these coming from, and it does feel a bit like a scavenger hunt, if you will, and that it's hard to put all the pieces together and figure out where it's coming from, and where the biggest difference can make. I get that question all the time when people read papers, or if we talk to press. Everyone wants to know, okay, that's great, you're finding that PFAS have all these kind of scary endpoints, but what do I do? How do I reduce exposure? Where is it coming from? Is it in my water? Is it in my food? I mean, where's the biggest impact? Is it that I get an air filter for my house? And is it the legacy PFAS that are a problem? Or, a lot of the water filtration systems say that they filter out the longer chain PFAS, but what about the shorter chain PFAS? Is our total PFAS concentration still actually increasing, to your point, because we have all these short chain or ultra short chain PFAS that are in our drinking water or wherever they are in our environment that we're exposed to? I think that would be really meaningful work, is kind of showing that where they're coming from, and how people can kind of like an intervention type study, how people can actually decrease, and how long does it take? How long does it take for someone to actually decrease internal and circulating PFAS? So yeah, I think that's needed.
Yeah, I agree that. Okay. All right. So I want to end this webinar with one last question. And then, so during your presentation, you showed that several PFAS and then weight change in teen labs study. And the very interesting thing was that PFNA and then PFDA compared to other PFAS. And as you mentioned that, other PFAS, there was a plateau in early time, and then the substantial decline after that. But these two PFAS compound, there was an increase in the weight gain, right? Was it something you expected, or this is okay, compared to the even longer chain PFAS compared to other? So any explanation for that finding?
Yeah, so PFNA and PFDA, I mean, it's a really slight increase, but it's essentially static. But there is a little bit of an increase up to that 6-month time point. I would say it's not on the scale that we're showing. It looks maybe like it's a little bit bigger than it actually is. But essentially, all of the PFAS basically are static.
Okay.
And then we observe a change at 6 months post-op, which I've given this paper a lot of thought. And my kind of thoughts are around just, you see improvement in liver function following bariatric surgery, right? So after a big weight loss event, the liver is kind of more productive. And so my thought is that you kind of see this delayed response, and then maybe the liver's filtering out more PFAS initially, and then there's the sharp decline. This is all just me kind of hypothesizing, but yeah, we see this kind of delayed response is how I think it's important to look at, rather than a sharp increase, where you would expect that with like a PCB or some of the other lipophilic pollutants. It's not quite that response, but there is a delay.
Okay.
Yeah.
All right. Yeah. Thank you very much for having this Q&A session as well. So we look forward to your, the follow-up study in the future, and hopefully we can invite you again in the future. Thank you very much. And then we have like in December, I believe December 2nd, we have the resident and researcher webinar, December 2nd on the MI course model, and then December 9 on artificial turf and children's health. And then you will receive this webinar announcement. And then thank you very much and have a wonderful Thanksgiving break, and then we will see you in December.
Thank you so
Bye-bye.
much for having me. Bye-bye.